started. Good morning. Welcome to the Centers of Excellence in Iron and Heme Disorders at the University of Utah. Today we are privileged to talk to Roland Lill, who comes from the University of Marburg. Roland is an expert in iron sulfur cluster biosynthesis and many other things, but today the interview is focused on giants in iron. So describing and discussing his experience as he moved through his research in the iron field and community. And we are joined by Cade Leverage, who is an MD PhD student here at the University of Utah, who will be conducting this interview. As a brief introduction for Dr. Lill, he got his, I guess you do call it a bachelor's degree?
A diploma.
He got his diploma in biochemistry.
Yeah.
At the University of Ulm and Munich. Is that both of those places?
Yeah.
Okay, and then he did a dissertation at the University of Munich in what I have up is German, so you're going to have to help me with this. Characterizing tRNA with the ribosome
interaction
and E. coli.
Absolutely correct, yes.
Okay, thank goodness.
This was about the third tRNA binding site in those days.
Cool. Okay, and he then went on to a postdoctoral fellowship in the laboratory of Bill Wickner while Dr. Wickner was at UCLA studying yeast genetics and vacuolar fusion events, which is near and dear to my heart. And then went back to Germany where
I still did, sorry to interrupt you, but I still did E. coli work, the SEC mechanism, SEC-Y, SEC-A, and so on. I discovered the SEC-A ATPase in Bill's lab, yes.
Okay, thank you for the clarification.
No
I didn't appreciate that. Roland then moved back to Germany and has been a professor since, if I've got this right, 1996?
Absolutely correct.
Okay, at the University of Marburg. And Dr. Lill has lots of accolades associated with him, and has been and is at the forefront of iron sulfur cluster biology, making seminal discoveries in there, and we'll hear a little bit more about that today. We'll turn the time over to Cade to conduct this interview. And Roland, thank you so much for being willing to participate. This has been a joy for me. And I know for Cade, it's been fun to meet the giants in iron who really established a lot of what we know about iron metabolism and homeostasis. So thank you.
Thanks for having me.
Yeah, like Diane said, I'm very excited. Me and my lab are very big fans of your work, and we love iron sulfur clusters. So it'll be great to hear about it. But before we get into the science, I'd just love to hear about you, your background and your family, kind of how you got into science.
Yeah. So I don't know how you define family. Do you mean the family I come from or my own family?
Yes.
Yeah, yes to both. Yeah.
Yes, then I have to start very early. As you know, I was born in 1955, and my father was a member of the so-called lost generation. At least we call it here. Lost generation is an expression for actually American writers like Hemingway, but we adopted this expression for those people who had to go to war, a Second World War, as young kids. My father was 16 when he had to go to war. When he came back, he was 17, so he couldn't go back to high school, and was basically convinced by his parents to go to do really bread and butter work. So he became a miller. And he never really wanted to do that. His dream was going to the United States and learn management. But he became a miller, married the miller's daughter, my mom. But he was one of the first to quit and sell all the milling machines, and actually without any formal education went into a management job. And after a few years, and we had to move 7 times until I was 18 years old, He had like 3,500 people under him in his social company. Pretty impressive. I didn't realize it when I was young, honestly. I only realized this tremendous effort and eager of my father later on. But I think I inherited it somehow. Coming to my own family, I have 3 kids. And we're a little bit special in that sense that my youngest daughter, the youngest daughter is already 32, has the Down syndrome. And of course, many people say, oh, how bad, how sad. But I can say she's the center of our family, basically the heart of our family. When my 2 older sons come home, they first ask about Sandra and not about the parents, which is absolutely fine and great.
That sounds familiar to me. I have a 6'2" youngest daughter. So she's quite tall, and nobody asks about anybody else, but everybody asks about Mary. With her unique sort of physique piece, not the mental challenges of Down syndrome, but I'm not sure actually people know my name often, but they absolutely know Mary's name. So I understand that family special child piece.
Yeah. And the latest thing is since 3 weeks, I'm grandfather of a baby girl.
Congratulations.
Congratulations. That's very exciting.
Very exciting, yes.
Yeah, best thing ever. Kate, how many kids do you have?
I've got 2. I've got a 6-year-old and 2-year-old. So
I tell you, grandparenting is magic. It's really great stuff.
That's what I've heard.
Congratulations, Roland. That's awesome.
Thanks.
Yeah, so coming from, that is a really cool story about your father kind of doing his dream without any formal education. That really is amazing. I guess the jump from management to science maybe to me is not clear, like the most logical progression, I guess. So like, how did that all come about? How did you get into biochemistry?
Yeah, that's also connecting me to my parents. The great thing about my parents was that they never said, do this or that. They always said, do something you like. And if you like it, then you can put in a lot of effort, invest a lot of time. Because, well, you will see your adult life involves competition with others. And you want to be good, you want to have a good life, and this means that you have to work. As a child, I never really understood that so well. And I once told my father, I will never ever work as much as you do. And during my PhD, he got me and said, I think you work very hard now.
With a big smile on his face.
Absolutely. And of course, when I went to the United States, his dream came true that, let's see, at least his son could fulfill a dream, go for a postdoc, even paid, to the US, to a real fun lab. This was one of my best times in life, also for my family with a young child, bringing one to the US, taking 2 home. My second son was born in Santa Monica. So this was really great. Yeah.
Cool.
Oh, sorry. Go, Diane.
No, you go ahead.
Oh, so I was just going to ask how you got involved in iron metabolism research.
Yeah. Well, this was a little bit of a detour. After coming back from the United States, I joined the lab of Walter Neupert, a giant in mitochondrial protein import, together with Jeff Schatz in those days. And I studied mitochondrial protein import across the mitochondrial outer membrane and got my habilitation. That's kind of a specialty in German science, to first do the habilitation before you get your own professorship. And before ending this, I felt I now have to develop my own research direction. And we figured, I also worked a little bit on apocytochrome C import into the intermembrane space, which is quite unusual because it uses a minimal machinery to go in there, to go into the intermembrane space. And we figured that the heme attachment, the covalent heme attachment with thioether bonds to apocytochrome C into intermembrane space in yeast and Neurospora involved two genes, or actually only one, a cytochrome C heme lyase and a cytochrome C heme lyase, two genes. But they are very similar, doing it for cytochrome C and cytochrome C1. And in bacteria at the same time, this was also something I got in contact with Sabia Merchant. I met her first at UCLA, and she's a big friend of mine still. I just met her here in Germany. And in bacteria, people had found 10 or 12 genes doing this job, attaching heme to C-type cytochromes in bacteria. So my idea was, okay, in yeast, we can find more of these genes. If we do a genetic screen, we did a genetic screen, we found an ABC transporter called ATM1. You may know this name. And we were pretty much excited. Hey, cool, we have the heme transporter. But soon we realized this is not correct because heme is made in the mitochondrial matrix, as you know, and we thought it's exporting heme into the cytosol. But when we overexpressed catalase in the cytosol, a heme-dependent protein, the catalase was just fine. Yeah. So we knew, oh, this is not a heme transporter. Same is actually true in human cells. We can overexpress catalase and deplete ABCB7. The catalase is still fine. And then we had a suffering 2 years when we did not know what is going on, but ATM1 depletion filled the mitochondria with iron. So we knew something is going on with iron regulation. And I always kept the eye open. But let's say decisive experiment came when I took serious the leucine oxytrophy of our TS mutant, which we originally had identified. And we analyzed the first, second, and third step of leucine biosynthesis. And you can imagine this as the first, second, and third step of a citric acid cycle. That's more well-known than the other leucine biogenesis steps. The carbon backbone is a little different, but the enzymes are very similar. And the second one, an enzyme called LU1 in the cytosol, was absolutely dead in the ATM1 TS mutant, and the first and third one were absolutely normal. And when I saw this result from a PhD student, she looked at me and said, I have this funny result. I have reproduced it already. I cannot get the second enzyme to work. Something is wrong here. And she saw my tears coming down of my face, literally, and says, is something wrong with you? And I said, no, but you have discovered something big. And she still didn't really get it. And then I scribbled it down and said, this is my view. Of course, in this stage, it was very early and very courageous to make the model that ATM1 is involved in cytosolic and then later on nuclear iron sulfur cluster biogenesis, which now is well established. Yeah. And that's, let's say, how we stumbled into the iron metabolism. ATM1 and mitochondria are a regulator of iron metabolism, not only in yeast, also in human cells, it's important. And we discovered, let's say, basically by this experiment, eukaryotic iron sulfur protein biogenesis.
And from there, you just kept rolling with it.
Yes, yes. And we sent this paper, actually, this is also a funny story. We sent this to Cell, and it came back after a few days because they said, the science is nice and it's great, but it's not important enough. And the first author, Gyula Kishpal, was, of course, very angry. I said, they didn't get it. I mean, we discovered something so important and so new. And I said, honestly, they will regret it.
Yeah,
And the
that's
paper
a
now
pretty.
has more than 800 citations, yeah, as far as I remember. Yeah.
It was so seminal. The discovery was just started that really, I
Thank you.
think, started that whole
Yeah.
field of research and trying to understand and
Yeah.
identify the machinery.
Right. And I should say, shortly after our paper came out, two papers came out from Val Kalodda and Betty Craig, who also discovered factors in iron sulfur protein biogenesis. Very nice. And I'm so happy that we are nice and friendly colleagues and even friends.
Yeah, yeah, absolutely. Jerry Kaplan still keeps in contact with Val Kulata quite a bit. Says she's doing well. So she might be an interview in a later episode for us.
Okay, I will look at
It'll be fun.
it. Yeah.
So along that line, I wanted to ask, making like these really foundational discoveries around iron metabolism, especially in the mitochondrion, I guess, what was it like in the iron field as you were making these initial discoveries, and how did that evolve throughout your scientific career?
Well, I told you already, I came from the protein trafficking field, protein transport field, and I was kind of competitive. Big heroes are in that field. I mentioned Walter Neupert, Jeff Schatz in the mitochondrial field, of course, Jim Rothman, Randy Sheckman, the Nobel laureates, Günter Blobel, and many others, just to name the people which were the most relevant at this stage. So this was sometimes really tough. And when I entered the metal field and went to the first meetings, I thought, well, the metal people, they're much more friendly to each other. They're competitive in a way, but also cooperative. They enjoy much more cooperation than competition. They rather do things together than secretly in their own lab. This, of course, didn't decrease, let's say, the rigorousness of science and the quality of science, but it was honestly much more fun to do it in a collegial way, right?
Absolutely.
Yeah.
I think that's the part as I age that I miss the most is sort of those interactions, because the iron community has historically been so collegial and helpful. Everybody rise, everybody go up together, and
Yes.
nobody get squished. So
there are some exceptions, but
yeah.
yeah, as always. Yeah.
Yeah.
No, I remember if I stay in Salt Lake City, of course, I remember a meeting in Sorrento. I think, yeah, this must have been the BioIron meeting in 2000, yes.
Yeah.
And I ran into, we already had published our EMBO paper, and we knew that Jerry Kaplan, a guy we followed the literature of, had published a paper on frataxin, and he had seen the same iron accumulation. In those days, we didn't know that frataxin plays a role in iron sulfur cluster biogenesis. This was just one possibility for the function of frataxin. And I remember the scenery when he came out of a door and he looked at me. Apparently, he knew my picture or something, and he said, "This is the guy whom I want to meet. I'm Jerry Kaplan." And this started a nice friendship. Yeah. So this was just wonderful. Something you never ever forget. Yeah.
Yeah. Just and great colleagues helping each other succeed.
Yeah.
So.
And of course, I should mention Sandy. She was also with him.
That's correct. Yep.
Are there any other influential mentors or colleagues that you want to shout out?
Absolutely, many. I should mention, let's say, that I was lucky to have three mentors in the early stage of my career. My PhD advisor, I mentioned, or Bill Wickner was mentioned, and Walter Neupert were mentioned. And they are very different scientists. So they approach science in a very different fashion. There are different personalities. Walter Neupert and Bill Wickner, they were big friends. Walter Neupert has died already, sadly enough, in 2019. But they were big friends despite they were completely different people. And I think this was absolutely a fantastic thing that I could take out of these researchers, let's say, what fit my own personality best. And so I can, advise younger people in the way that they should look at, let's say, older people, how they do it, how they perform their science, and they should simply pick what they like and leave out what they don't like or maybe even dislike. Because some things they just don't fit to you. And if you communicate with older people, I think you can learn just a lot, not only, let's say, scientific facts, but also how you perform, how you, for instance, simple things, how you give presentations. And I usually told my PhD students or postdocs, look up, if you don't understand or if you're not interested in a presentation, then at least watch this person, what he does in a cool fashion and what you don't like, and then try to learn from this what you should do and what you should not do. And I think that's a nice trick. And it doesn't cost anything.
And good
No
advice.
training course.
Yeah, that is good advice. What about some of your most influential trainees?
Yeah, I just don't want to mention also, if I go back a little bit, I just don't want to mention, let's say, only my mentors. Sorry for going back now to this, the first, or this previous question. I think in the metal field or in the iron sulfur field, I have many friends. And if I mention a few now, I think I leave out many. But I would like to at least mention from the early times, the people whom I still talk to is Dennis Dean, because his paper in JBC in 98 got us going to see, okay, the ISC machinery present in bacteria is also present in mitochondria. This was very influential. It came out when we wrote up our EMBO paper I talked about, and the NIF-S idea basically was already present in our lab. We had studied this enzyme already, but the NIF-S or the ISCS in the bacterial operon created the idea in our lab that, let's say, all the operon genes or proteins should be in mitochondria. And so not only we had ATM1 and NIF-S in our first paper, but I think, forget, nine other suspected, proposed ISC proteins, and they were all correct even. Sometimes if you make such suggestions, there is one guy off, but they were correct. So Dennis Dean, and I was also impressed, and the person left the field too early, Larry Vickery is also a very great person. And of course, I mentioned Jerry and Dennis from Salt Lake City, Jerry Kaplan, Dennis Wingy, who became very good friends. Dennis also a biking friend. Dennis visited me here in Marburg for 4 months as a little sabbatical, which he spent here over in Germany, which I think he enjoyed a lot.
I actually can confirm that. I know he enjoyed a lot. Yeah.
Yeah, we had a great time.
That's terrific. Also a few little trips to Zion here and there, where a good crowd of us had a lot of fun together.
Yeah, and White Room. Yeah. Yeah, you asked also about, so I don't go back to, let's say, the iron sulfur field. There would be so many wonderful colleagues I could mention, also collaborators I could mention. Maybe the time is not allowing this. You were starting also to talk about trainees. I mentioned one already, and this is, of course, when I only talk about the iron sulfur field and not the early time in Munich. This is, of course, Gyula Kishpal. He was a wonderful scientist, very excellent experimentator. Sadly enough, in a car accident in 2003 already, he died together with his father in Hungary. And we collaborated still, and I still can't believe it that he's now not with us anymore for 21 and a half years. So basically every year we light up a little candle on his birthday to remember him and think about him, because he was starting my career. I would also like to mention Uli Mühlenhoff and Oliver Stelling. You possibly know the names from our papers. These are staff members in my group, and I think without them, our science wouldn't have been the same. They are excellent scientists. I had many excellent co-workers, PhD students. I just want to mention, for instance, Heike Lange, Jannike Balk, Rafał Dudkiewicz, Antonio Pirig and Dailinets, and Vasundra. They all have staff positions in various institutions. Did I mention Rafał Dudkiewicz? He's in Gdansk now. And I want to mention also a very good scientist who went into a different career, not into academia, unfortunately, Alex Scheftel. He was an excellent postdoc coming from Prem Ponka's lab and went back. And he was very unfortunate with his postdoctoral time back home because his project didn't work out, and at least the first project. And then since he had family, he gave up and went to industry. I still suffer a little bit that this excellent person didn't stay with us in science, because I know he enjoyed it so much to be in science.
That's pretty sweet memories of contributions of trainees, Roland. I
could mention many more, but of course,
yes, but I love that. I love your tender memories of each of them. Yeah.
Yes.
Yeah, I agree. Thank you so much for sharing that, and really everything you've shared. You've mentioned a few pretty fun stories about interactions with people in the iron metabolism field. Is there any other fun story you feel like would be another good addition to this interview? If not, it's okay. I have more questions.
No, I think I can come up with one. And I come back to Sabia Merchant. We were basically after lobster speakers in a GRC. I think it was cell biology of metals, or maybe it was mitochondria and chloroplasts, I guess, right? So everybody had drunken wine, had eaten the lobster, and they were in the lecture hall, and Sabia had to give her presentation before me. And this was still during the times of transparencies, and Sabia is kind of short. She hardly made it up to the transparency surface to put up her transparencies, and the crowd was always making fun of her. And of course, she enjoyed it a lot. It was a very good scenery. Then I was next, and I presented the first reason why mitochondria are really essential, not because of respiration. They are not essential because of respiration, but because of iron sulfur cluster biogenesis. And the protein in the cytosol, which I introduced, was RLI1. And then I usually in my presentations this is an iron sulfur protein in the cytosol. I made the joke, people in my lab call it ROLI1. And then a friend of mine was yelling at me, is there a ROLI2? And then before I could answer anything, someone else, also a good friend of mine, Rosemary Stewart, said, one ROLI is enough. And then, honestly, for 2 or 3 minutes, I couldn't continue my presentation because everybody was laughing and tears came down. Oh, this was so funny. Yeah.
I love those kind of memories.
Yes.
Yeah, that's it. That's another excellent addition.
Yeah.
And so I've got another kind of loaded question for you, which I
Mhm.
enjoy asking these questions. You know, your scientific career has encompassed a lot of really foundational discoveries. But if you had to pick one or two, um, what are kind of the discoveries you're most proud of?
Mhm. One or two. I mentioned it already, the ATM1 discovery, because this started the field of iron sulfur protein biogenesis in eukaryotes. I think this was the most seminal single piece, but I would also like to mention a few other papers which I'm really proud of because it ordered something in the field. That's, for instance, Uli's EMBO paper in 2003. We call it a staging paper, where we actually staged the various ISC components in a kind of a linear sequential fashion. And at that stage, the in vitro data were sometimes a little bit confusing. And we used in vivo data to say this protein is first, then this is second, this is third, and then the cluster is inserted. And this is still correct. And I think people use this model now, and I think this was kind of an important thing. If I come, let's say, to the recent things, I'm basically retired now. Then I'm kind of proud that in the end of my career, we could get structural insights and mechanistic insights in how a cluster is actually synthesized. Get, for instance, down what the function of frataxin is. You can actually see it in the various crystal structures, how ferredoxine puts its electron into the ISQ2 assembly site that can now be seen. This is not the end of the line. There's lots to be done. But I think this started now, the mechanistic molecular understanding of how a cluster is actually made. Yeah.
Well, that leads perfectly into probably my favorite question to ask, is, in your mind, what is the biggest discovery to still be made in the field of iron metabolism?
You mean my discovery or general?
Or what's your biggest question, unanswered question in the field? Like what kind of like keeps you up at night thinking about?
Well, that's still, I would say, in a single mind, it's still the ATM1 substrate, because that's still not clear. From physiology, and we did a lot into this direction. Other people did a lot into this direction. From the physiology, I think it's now consistent. We don't know, but it's consistent with what we originally, when we were young and courageous, proposed. It may be a 2-iron 2-sulfur cluster shielded with glutathione. I can't believe so well that, let's say, glutathione only is basically the plus a 2-iron 2-sulfur is the substrate, because I don't know how ATM1 could pull out this entity from the solution and be a good competitor against 10 millimolar glutathione swimming around in the mitochondrial matrix, right? So in terms of affinity, the glutathione and this compound, which was synthesized by Jimmy Cowan, the tetraglutathione for 2-iron 2-sulfur cluster, the binding constants apparently are not so different that I could believe that this is going on. So there must be something else in addition. But I don't know. We have tried to trap it in the cryo-EM structures, but this so far hasn't succeeded. There's a paper out where some density in the ATM1 is in there from a Swedish group, but they couldn't decipher, resolve what the molecule actually is. So that's a big question for the future. And then, of course, other questions are what is the mechanism of the individual steps? And I think so far people have thought of this protein, that protein, and so on. And I think the better idea, also combined with cryo-EM structural analysis, is to look into complexes, how complexes, many proteins function together to support a particular step. And I think if you now do cryo-EM, if you're lucky, and we were lucky sometimes, then you can get several different structures from the same grid, basically, and see how the mechanism, or get at least suggestions how the mechanism could be. And this provides you a testable, biochemically testable idea on how it could run. And then, let's say, by making mutants, doing intelligent biochemical experiments, I think you can see or suggest what the molecular mechanism actually might be. I mentioned the core ISC studies, a whole series of papers. Most of them were in Nature Communications. And another one is just accepted now with the ferredoxine structure. And I think this is basically the new way to go and understand the mechanism in each step of this complicated process.
Especially for those that, I mean, it's been years that people have tried to work out this biochemistry in vitro, and it's been hard. And so these crystal structure pieces are
Yep.
just so essential, because
Yep.
they then give you guidance on, like, you can go create mutants and things like that and confirm. And it's just been essential.
That's what we did with the help of these crystal structures. A crystal structure or cryo-EM structure is just a start. It's not the solution, because you also have to be careful not to overinterpret the structures, because there are sometimes snapshots and could be dead ends in the row. So you have to be careful in interpreting them. But the biochemistry then will tell you whether something you see in the structure is important or not. And not only the biochemistry. One thing my group always did, I always tried to do that, is to combine. I'm a biochemist, trained as a chemist actually, but I moved more and more into biology. And whenever I have a phenotype in a cell, I think that's exciting, because then I know whatever I did to the protein of interest, if this creates a certain phenotype which can recapitulate what I see in a test tube in vitro, then I'm a little bit closer to truth, right? And you will, of course, never reach truth. And that's why I would like to mention this, if I may. The mitosome story for us was so exciting. Mitosomes, for people who don't know this, are cellular organelles which are similar to mitochondria, but as opposed to mitochondria, they are evolutionarily reduced. These organisms containing mitosomes, they have lost many genes, more than 1,000 genes, which you usually know are mitochondrial, encoding mitochondrial proteins. They're all gone, apart from a few proteins which make the mitochondria, Tom and Tim protein translocases, some chaperones, and the only biochemical process which is left over in these mitosomes is iron sulfur proteins, IC protein.
As you might predict.
As I had predicted, and when I came across this, I said, damn thing, apparently my biochemical, courageous idea in the EMBO paper I mentioned is probably not too incorrect. This is honestly the biological proof, because the mitosomes, they don't even have their own iron sulfur proteins, right? They lost even iron sulfur proteins like aconitase or something like that.
Yeah, really foundational, actually, in confirming that.
Yeah, I agree. I agree. And I've really enjoyed this interview. And I guess I just want to close it out just asking for young scientists like me, both interested in iron metabolism, but also just wanting to carve out a niche in science. What advice do you have as we try to make our own important discoveries?
What advice for you? Well, I think I cannot give you advice as part of the metal field or iron field. It's a general advice. And I think this goes actually back to what my father taught me, or my parents taught me, do what you enjoy. I think in science, we have the privilege to actually do it, with some restrictions to get money in for the lab, of course. But do follow your own interests, follow your curiosity, try to do something new, explore something challenging, even if people say, this would never work. You may get it to work. You always, if it doesn't work, you can put it on the back burner and do something else, but maybe it comes back and you remember that you have something on the back burner, and then you're a little bit more knowledgeable and you can put it to the front and maybe it works. This happened to me several times, that I had things on the back burner, left them for a while, and after a while they were coming back. Of course, what you also have to see is not follow your own ideas just by having your own fun. You also have to look a little bit that you excite other people with your ideas. Because if you excite other people, then you get reviewers accepting your paper, you get reviewers giving you money in grant applications. So you have to always look a little bit what other people think about your ideas. But if you, let's say, follow new routes, try to discover new things, I think you're on the right track, because then you can always thrill colleagues because you do something what they don't know yet. Yeah.
Well, excellent. Thank you for that. Is there anything else you wanted to add, Diane? Any questions you wanted to throw in there?
I don't have any questions. I just want to pitch out there a raw for Roland as a leader in the field, but also some people might not know and might give the name as an individual being a goat because they kind of are a leader in the field. Literally, Roland Lill is a mountain goat, if you have ever hiked with him.
Absolutely, yes. I love hiking, yes.
Yes. And literally kind of an elevator guy. If you're
Yes.
going on a hike, Roland has no problem going up.
The problems start.
Sorry. Anyway, just
Yeah.
a goat in all the senses of the word that is a current popular term.
Yep. But my recent vacation with my wife was, of course, again in the Alps.
See? I knew that would be true.
Yeah.
Roland, it's been just terrific talking to you. This has been really fun, a great sort of walk down memory lane of discoveries. It makes me want to pull out a whole bunch of literature and go read it again, because I know I'll learn new things that I didn't think about. And I really appreciate that. And
thanks. It was also fun for me.
Yeah.
And I hope we'll see each other soon.
Yeah, that would be wonderful. I hope that we're doing these recordings and we're going to post them on our website, but also the Utah Center for Iron and Heme Disorders has a YouTube channel. So they will also go up there for a broader audience. But I'm going to try and compel maybe the National Library of Medicine, at least here in the United States, to try and archive these interviews, because I just think there's so much history that's important in young people's careers, learning from those of us who we would call senior in the field. Yeah.
Yeah.
So this has been really fun. Thank
Can
you
I
so
add
much.
one more name?
You bet.
Oh, I forgot this name. If I'm correct now, I'm very proud of having been introduced to Helmut Beinert. And of course, Helmut is a wonderful scientist, a wonderful person. And we invited him to Marburg several times because other colleagues knew Helmut from other stories. And he reviewed my first PNS paper on iron sulfur cluster biogenesis, corrected my English. And I'm very happy and proud to say that he loved our science. I was even at his house enjoying dinner with him. And he's still one of my heroes because he's not only a great scientist, he started his career as an actor. Hardly any person knows, as an actor here in Germany. And then, of course, he was another victim of Second World War because the Americans basically offered him a position in the United States when Germany was completely devastated and he couldn't do research anymore. So he escaped from our country and we lost him. So you see what wars do to people. And honestly, 70, 80 years later, we are not smarter than in those days, if you look into different parts of the world.
That's a great ending. Thank
We
you
should
so
be
much.
smart.
Yeah. Thank you so much, Roland and Cade. Great interview. And I'll let you know when we do get all of this posted, so that you can go have a look at everybody else's talks too, because they're quite fun.
Yeah. Thanks, Cade. This was fun talking to you. And of course,
Same.
you
It was
know
wonderful to
Diane.
meet you.
Okay. Take care.
And greetings to Paul and the rest of the crowd.
I will pass that on for sure.
Yeah,
And Cade, you
absolutely.
pass it on because you're just down right there, out the door.
Will do. Will do.
Okay. All
Okay,
right. Take
bye-bye.
care, everyone. Bye-bye.
Bye-bye. Thanks a lot. And
Yeah.
thanks for having me.